Have you ever really stopped to think about how a creature so small, I mean, you can barely see it, manages to make these incredibly precise decisions, like life or death decisions, all based on its environment?
It's a fundamental question, really. How does any organism, tiny worm or, us, perfectly tune its behavior to what's going on around it?
Exactly. And today we're taking a deep dive into that very question, focusing on the humble roundworm C. elegans.
Mm.
We're going to uncover the intricate neural circuit it uses to intelligently decide when to lay its eggs.
And it's all about food availability. It's quite remarkable.
It really is. Our mission today is to unpack this specific reproductive behavior because honestly, in doing so, we uncover some really fundamental principles of neuromodulation.
Principles that apply far beyond this microscopic creature, for sure.
Right. And this deep dive comes from a really insightful research article, plus your notes, giving you a shortcut to being genuinely well-informed on this amazing system.
Let's get into it.
Okay, so let's set the stage. We've got these C. elegans hermaphrodites.
Meaning they have both male and female reproductive organs, yeah.
Right. And they have this super clear, very strategic approach to reproduction. They release fertilized eggs when there's plenty of food around, like a good patch of bacteria.
Makes sense.
Provide for the kids.
Exactly. But, and this is crucial, when food is scarce, they hold on to those eggs. It's a pragmatic choice, purely about survival.
And the evolutionary stakes here, they are incredibly high. If that food deprivation goes on too long, the mothers will actually keep the eggs inside until they hatch internally.
Whoa.
Yeah. And this process, it often kills the mother. So you can see why having a really accurate, finely tuned food sensing mechanism is just absolutely vital for the species.
It's intense, and you realize these tiny worms are making decisions with stakes just as high as, any big dramatic human dilemma, just microscopic.
Absolutely. The scale is different, but the weight of the decision isn't.
Okay, so we actually know a fair bit about the core mechanics of the egg-laying system itself. It's surprisingly complex for such a small animal.
It is. You've got 16 muscles involved. 16. And 4 motor neurons, including 2, the serotonergic HSNs, that are just indispensable. They're the direct activators, right?
They're the ones telling the muscles to contract, yeah. Plus, you have neuroendocrine cells, like UV1, releasing inhibitory stuff like tyramine.
Which helps sort of calm things down after an egg is laid?
Exactly, resets the system. But while those core parts were known, the real puzzle was how food availability modulates all this.
Because it's not just one food sensor.
Not at all. C. elegans uses this incredible diversity of food sensing mechanisms. Gustatory neurons for taste, olfactory for smell, tactile for touch, all detecting bacteria.
Wow.
And even interoceptive neurons, sensing metabolites or bacteria inside the worm. It's like a whole symphony of sensory inputs converging on this one critical behavior.
A symphony. That sounds incredibly hard to untangle. So where did the researchers even begin? How do you pull on that thread?
Well, their starting point was kind of clever. They looked for genetic suppressors.
Suppressors.
Yeah, basically other mutations they could fix or suppress the problem seen in a specific mutant called AG6GF.
Ah, the AGL6GF mutant? That one became a key clue, didn't it?
It really did, because it showed what happens when the system gets stuck in the off position, essentially. This mutation amplifies an inhibitory signal onto those HSN motor neurons.
The ones that trigger egg laying.
Right, so this extra inhibition causes the worms to just bloat with unlaid eggs. It's a very clear sign that their egg laying is jammed.
So finding something that could undo that bloating, that
Yeah.
would point towards the missing regulatory piece.
Exactly. It's a powerful way to find new players in the system.
Mhm.
It really shows how even in simple systems, tiny imbalances cause dramatic problems.
And this is where the accidental hero comes in, the N5210 mutation.
That's the one. A single lucky find. This mutation profoundly suppressed that bloating in the EGL6GF worms,
And it fixed the HSN activity, too.
almost back to normal levels. Though, interestingly, the HSN spikes themselves looked a bit different, narrower, faster decay. Subtle, but significant.
Okay, so what is N5210? What kind of mutation is it?
What's fascinating is its nature. It's a gain-of-function mutation.
Meaning the gene is overactive.
Correct. And the gene is 2WK17. This gene codes for something called a two-pore domain potassium channel, or K2P channel.
T2P potassium channel, okay.
Think of these like tiny dimmer switches on a neuron's activity. They help control how excitable it is.
Right. And a gain of function, an overactive version of a channel like that, what does that usually do?
It usually leads to neuronal silencing. effectively dimming the neuron's activity way down.
Okay, so they found this N5-10 mutation, which is a gain of function in 2WK17, and it fixed the egg-bloated worms. So the next step, see what it does on its own.
Absolutely. Take it out of that AGL6GF background and see its solo effect.
And the results were revealing, I bet.
Very revealing. When they isolated the TDK17GF mutation, it completely messed up the worm's ability to stop laying eggs when food was gone.
Ah, so it broke the modulation part.
Exactly. These mutants just laid eggs pretty much constantly, whether food was there or not. Totally unlike wild-type worms, which mostly stop laying without food. This was a huge clue they were onto something central to the food-sensing modulation itself.
And to really quantify this, to put a number on it, they came up with the ELMO index.
Yeah, the egg-laying modulation index. Brilliant little metric.
See, how did it work?
Simple and clear. Wild-type worms, the normal ones, had an ELMO index close to 1, meaning, they laid almost all their eggs when food was present.
Makes sense.
But the TWK17GF worms, their index was near 0, laying eggs pretty much equally, food or no food.
That really nails it down. It shows TWK17GF isn't just affecting general egg laying, it's specifically disrupting the modulation by food. And it's acting separately from that EGL6 pathway.
Precisely, a parallel pathway, crucial for this regulation.
Okay, so TWK17 is a key player. Where is this channel actually working? Which neurons use it?
Right, that's the next big question.
And also, how does making a potassium channel overactive, which usually silences neurons, lead to more egg laying in the absence of food? It seems counterintuitive.
Good point. So they looked at expression patterns, and TWK17 turned out to be prominently expressed in a specific pair of interneurons called AVKs.
AVKs. Okay, where are they?
Their cell bodies are up in the worm's head, but their extensions, the neurites, run down the ventral cord. Right near the egg-laying system, actually.
Convenient location.
Very. And as we said, KTP channels like TWK17 are negative regulators, dimmer switches. So that gain-of-function mutation in TWK17 would effectively silence these AVK neurons.
Okay, let's unpack that. If silencing the AVKs is the effect of the mutation, does artificially silencing just the AVKs cause the same problem? Does it mimic the 2WK17GF defect?
Yes, that's exactly what they tested. They specifically expressed the mutant 2WK17GF channel only in the AVKs, and it perfectly recapitulated the egg-laying modulation defect. Bingo. They even went further. Used fancy tools like chemogenetics, using histamine to silence neurons, or optogenetics, using light to activate them.
So they could turn AVKs on or off, precisely.
Exactly. Silencing AVKs caused that strong modulation defect, just like the mutation. But activating AVKs? That was surprising.
What happened?
It actually suppressed egg laying, even when food was available.
Okay, so AVK neurons are sending some kind of inhibitory signal to the egg-laying system. Silencing AVKs removes that inhibition. Activating them cranks it up.
You got it. That's the core idea.
But how do the food cues themselves control the AVKs? What's the input?
Right. So they measured calcium activity in the AVKs. That's a good proxy for neuronal activity.
And what did they see?
When food was present, AVK calcium was stable and low. Pretty quiet. But take the food away.
Let me guess, they got more active.
Exactly. AVK calcium levels went up and started fluctuating. This showed clearly that AVKs are normally less active when food is around.
Suggesting that some food sensing mechanism must be inhibiting the AVKs when food is present.
Precisely. Inhibiting the inhibitor allows egg laying to happen.
So the hunt is on for the neurons that sense food and then inhibit AVKs. Any prime suspects?
Well, previous work and anatomical maps pointed strongly towards the PDE sensory neurons.
PDE neurons, what's special about them?
They are dopaminergic, they use dopamine, and crucially, they're known to be directly activated by food stimuli. Plus, they make lots of synapses, connections directly onto the AVKs.
Dopamine, food sensing, connection to AVKs, the pieces are fitting together, but they needed that smoking gun, right? The experiment to really prove dopamine was the messenger.
Optogenetics again provided the key insights. It's such a powerful tool.
What did they do?
First, they inhibited the dopaminergic neurons using light. And guess what? Egg laying went down.
Okay, less dopamine, less egg laying.
But here's the kicker. This effect was much weaker if the AVKs were already silenced by the TWK17GF mutation.
Yeah, suggesting the dopamine signal works through the AVKs. If AVKs are already off, inhibiting dopamine doesn't do much more.
Exactly. Then they did the opposite. They activated the dopaminergic neurons with light. That triggered egg laying, even when there was no food around at all.
Wow. So activating the food signal basically overrides the lack of food.
And critically, this effect depended on a specific dopamine receptor, a D2-like receptor called DOP3. And guess where DOP3 is expressed?
Let me guess. The AVKs.
Bingo. Specifically in the AVKs.
Okay, pull it all together for us. What does this whole dopamine story mean?
It means that these dopaminergic food-sensing neurons, the PDEs, promote egg laying. And they do it by directly inhibiting the AVK interneurons using dopamine acting on the DOP3 receptor.
So it's disinhibition. Food activates PDEs. PDEs release dopamine. Dopamine inhibits AVKs.
And inhibiting the inhibitory AVKs takes the brakes off the egg-laying system. Food present equals foot off the brake.
That foot off the brake analogy is perfect. It really clarifies the logic. Okay, so AVKs send an inhibitory signal. How do they send it? Not with typical fast neurotransmitters, you said?
Right. AVKs seem to rely heavily on neuropeptides. They express a whole bunch of different neuropeptide genes.
And they have the machinery to release them.
Oh yeah. Rich in all the factors needed for synthesis and secretion of these peptides. It strongly suggests neuropeptides are their main language, more like a slower, broader broadcast signal.
So how did they prove that neuropeptides from AVKs are essential for this food modulation effect?
A couple of clever experiments.
Blah.
First, they blocked neurosecretion specifically for AVKs. They used tetanus toxin, or they knocked down a gene called UNC31, which is essential for releasing these neuropeptides.
And what happened to egg-laying modulation?
It caused a profound defect, very similar to silencing the AVKs themselves.
Okay, so blocking the output signal breaks the system. What about boosting the signal?
They did that too. They used a genetic trick to upregulate neuropeptide release from AVKs. That inhibited egg laying, even when food was present. It basically locked the brakes on.
Okay, definitive proof. Neuropeptide release from AVKs is central to inhibiting egg laying in response to the absence of food. So which peptides? Is it just one, or?
Not just one. That's another fascinating layer. Through huge genetic screens and looking at gene expression, they narrowed it down to 3 key neuropeptide genes from AVKs that seem to work together.
3 working together. Which ones?
PDF1, NLP10, and NLP21. Mutating them, especially PDF1 and NLP10 together, caused really strong modulation defects, almost as strong as silencing AVKs entirely.
So AVKs send out this cocktail of neuropeptides, PDF1, NLP10.
And they know the receptors for those two are PDFR1 and NPR35, respectively.
A compound message, then, not a single signal. Where does this complex message actually land? Where does it converge?
That was the next puzzle. PDFR1, the receptor for PDF1, is pretty widely expressed, so they needed a more specific approach.
How did they find the specific target cells?
They used a really cool technique called intersectional genetics. Imagine shining 2 different colored spotlights, one for cells with the PDFR1 receptor, one for cells with the NPR35 receptor. They look for the cells where the spotlights overlapped.
The cells that have both receptors?
Yeah.
Smart.
Very smart. It acts like a highly specific target finder.
And which cells lit up with both spotlights?
The prime candidates were a small group of head neurons, but the most consistent and strongest hit. The AIY interneurons.
AIY interneurons.
Okay, and they proved it functionally. They show that restoring the expression of both receptors, but only in those neurons that normally co-express them, like AIYs, was enough to rescue the egg-laying modulation defects seen in the receptor mutants.
That's strong evidence. AIYs are the key receivers of this compound neuropeptide signal from AVK, but are they absolutely necessary?
Great question. They tested that directly. They used mutants called TTX3, which specifically lack functional AIY neurons. They just don't develop properly.
And what happened to their egg-laying modulation?
These TTX3 mutants showed modulation defects that were just as bad as completely getting rid of the neuropeptide signals from AVKs, or even silencing the AVK neurons themselves.
Wow. Okay, that seals it. PDF1 and NLP10 from AVKs act on AIY interneurons to regulate this whole process. Unequivocal.
Absolutely.
So, final step in the circuit then. How do the AIY neurons actually influence egg laying? What's their output?
The study proposes that when AVK neuropeptides hit the AIYs, they likely excite the AIYs.
Okay, AIYs get activated.
And then, in turn, the activated AIYs likely inhibit the downstream egg-laying motor neurons like the HSNs we talked about earlier, possibly using acetylcholine as their signal.
So wait, inhibition leads to excitation, which leads to more inhibition.
Exactly. Another layer of inhibition in this incredibly elegant circuit. It's like a carefully orchestrated chain reaction of inhibitory signals to fine-tune the final behavior.
That is just fascinating. What an incredible deep dive into this tiny worm. The sophistication packed into that small nervous system is mind-blowing.
It really is.
But let's zoom out. What are the bigger lessons here? The universal principles of neuroscience that C. elegans is teaching us through this specific example.
Yeah, there are at least three really critical features here that illustrate general principles found across pretty much all animal nervous systems.
Okay, lay them on us.
First, disinhibition as a fundamental circuit architecture. This study shows so clearly that the default state for egg laying is off, held back by inhibition from the AVK neurons.
Right, the brake is on by default.
Exactly. And food cues promote egg release by inhibiting the inhibitor silencing AVKs, thus relieving that default inhibition, taking the foot off the brake.
We see that elsewhere, right?
Everywhere. This disinhibition acts as a powerful gating function. You see it in the vertebrate basal ganglia controlling movement all over the place. It adds layers of control, regulation, plasticity, essential for adapting behavior. It's a fundamental brain strategy.
Okay, disinhibition. What's number 2?
Second is the power of combinatorial neuropeptide signaling. We often think of neuropeptides as one-to-one signals. One peptide, one receptor, one effect.
But that's not what's happening here.
Not at all. The AVK to AIY connection isn't based on one peptide receptor pair. It's specified by the co-expression in AIYs of two distinct receptors, PDFR1 and NPR35, for two distinct AVK neuropeptides, PDF1 and NLP10.
So the receiving neuron needs to hear both parts of the message to respond correctly.
Precisely. This combinatorial logic massively increases the potential complexity and sophistication of signaling within these neuropeptide networks. AVKs aren't just sending an on-off signal, they're acting like hub neurons, sending a complex, nuanced message.
Wow. That opens up a lot of possibilities for how neurons communicate. And the third principle?
The third is the role of K2P channels as key tuners of neuromodulation. This research really highlights how these potassium channels, like TWK17, aren't just passive leak channels.
They're actively involved.
Yes, they subtly regulate the excitability of neurons like AVK. They act as specific effectors for signaling pathways, crucial mediators of neuromodulation. You find them everywhere: sea slugs, fruit flies, humans. They're involved in learning, sleep, neurological function.
So they're like fine-tuning knobs on neuronal activity responding to specific signals.
Exactly. And they can even integrate metabolic or physiological signals. They might act as interoceptors in a way, linking the animal's internal state, how hungry it is, its overall health, directly to its behavior.
Which leads to a really intriguing idea. Maybe those TWK17 channels in the AVKs aren't just sitting there. Maybe other signals, other circuits or internal state signals are constantly adjusting the dimmer switch on the AVKs.
That's a fascinating possibility. It would dynamically tune how tightly food sensing is coupled to reproduction, allowing the worm to adjust its strategy based on its overall condition, not just immediate food availability. Like a master volume knob for this whole behavioral symphony.
What an absolutely incredible journey into C. elegans. From a worm deciding when to lay an egg, we've pulled out these huge universal principles: the elegance of disinhibition, the rich language of combinatorial peptides, the subtle power of K2P channels.
It just hammers home that you don't need a giant brain to have an incredibly sophisticated one. The complexity is truly stunning.
And understanding this neurochemical logic, as you called it, in a system you can actually map out like this.
It offers profound insights into how all nervous systems work, including our own complex brains. And this isn't just academic curiosity, right?
No, definitely not.
This kind of fundamental knowledge pushes forward our understanding of neurological conditions, psychiatric disorders. It could genuinely open up new avenues for therapies down the road.
So as we wrap up, what stands out most to you about this incredible neural dance? Maybe it makes you wonder, doesn't it? How many of your own fundamental behaviors, appetite, mood, sleep, who knows what else are being orchestrated by similarly elegant, maybe disinhibitory circuits, all fine-tuned by this complex symphony of signals happening completely below your conscious awareness.
It certainly makes you think about the hidden complexity controlling even our simplest actions.